Okay, this is only a little bit theory about the onset of effect, what really determines the onset. And we have a couple of different steps until you really see the effect of the neurotoxin. The first is the migration or diffusion to the motor end plates. So the neurotoxin has to find the receptor on the motor end plate on the neuron. And then we have this other steps, binding to the receptor. This binding process is very fast. So when the receptor meets the neurotoxin or the neurotoxin meets the receptor, in less than 2 seconds, there is a binding. So very fast process. Endocytosis is also very fast. These, what I've shown you, this opposite effect from the secretion. That means a secretory vesicle is formed and transported into the cytosol, into the inside of the cell. Very fast process. The translocation takes some time. This is also done in cellular systems. It takes about 20 minutes until the light chain is translocated into the cytosol of the nerve cell. And then in the end, there's cleavage of this SNAP25, of this protein, which is responsible for the secretion of acetylcholine. It's dose dependent, but because it's an enzyme, it's also very, very fast. So this is an interesting figure. 25% of this SNAP25 must be cleaved, and then paralysis starts. So altogether, what really determines the onset of the effect is this: the migration of the neurotoxin to the motor end plates. And the consequence is, the better you hit the motor end plate, the better you know where the motor end plate in the muscle is, which you must treat, the better is the effect. And the earlier is the onset, and the less neurotoxin you must apply. So knowing the anatomy is very important because of this migration effect. All the other steps are very fast, but the migration takes a long time, a couple of days. Okay, the other interesting effect that might be more interesting for you in the clinical practice is the duration of the effect. What determines the duration of the effect? Well, we have to consider that the light chain is a protease, an enzyme, which cleaves every newly synthesized SNAP25. And the cell is continuously producing SNAP25. The half-life is about 48 hours. So permanently SNAP25 is produced. But the newly synthesized SNAP25 is cleaved by the light chain which is present in the cell. So the cell produces, but the light chain cleaves. The other thing is that the cell produces proteases which degrade this light chain in the nerve cell. It is not known right now what protease, maybe it's the so-called proteasome, a special machinery in the cell, or the lysosomes or other proteases are responsible for the degradation of the light chain. But continuously the light chain is degraded so that more and more active SNAP25 is present in the cell. And in the end, there's enough 25, the cell has recovered, and you have to inject again to have the therapeutic effect. So that is the explanation for the duration of the effect. The duration of the light chain in the cytosol. From that, you can draw a conclusion. That means the concentration of the light chain you have in the beginning, at the maximum of the effect, really determines the duration of the effect. If you have the same concentration, the maximum effect, you have the same duration of the effect. If you have less, the duration is decreased. So that is the explanation. By the way, maybe that can be interesting for you, how the duration of the effect can be analyzed in animal system. This is the so-called mouse running assay, and we have here, or the scientists have here mice in this cage with a running wheel. And so these mice, they run at night, they are night active, and this is very interesting, they run about 10 kilometers per night. Really long distance, more than I usually run per week. And these mice, when they are trained for this 10 kilometers, they are injected into the gastrocnemius, in the lower leg muscle. And you can see here, dependent on the dose, you find a different maximum of inhibition of the running distance. Here 0.8 units, here 0.4 units. And this also determines, if you extrapolate that, determines the duration of effect. So with this system, you can really elegantly analyze the duration of the effect. This is not, well, maybe we should continue with this. And then we go to some clinical studies. Interesting effect with botulinum toxin, the so-called saturation effect. That means the following. You know from the action mechanism that botulinum toxin can only be taken up when the receptor is exposed. This molecule which is named SV2. So when there is secretion, this SV2 molecule is exposed, and then the new toxin can be taken up. And only when the SV2 is exposed, the new toxin is taken up by the cells. That means, in the beginning, we have a continuous secretion of acetylcholine, because we have spastic muscle, so to speak, and there's continuously exocytosis. The receptor is exposed. New toxin can be taken up by the reverse process. But then the neurotoxin inhibits the secretion of the acetylcholine, and it inhibits on the same mechanism the exposure of the receptor molecule. So when there is enough of the neurotoxin in the cell, there is no secretion anymore, and that means no SV2, no receptor is exposed anymore. That means no further uptake can take place. So the neurotoxin inhibits, so to speak, its own uptake. That means that above a threshold, above the saturation threshold, you can inject and inject, but there is no more effect because of the saturation. Interesting effect is shown. I think I have an old slide. This is just reading from the EDB assay. So in the extender divitum brevis, in the small toe muscle, botulinum toxin is injected, and with a couple of these are electrophysiological measures which show you the paralysis. It is analyzed whether there is an increase of the effect in the paralysis. And as you can see here, you find a saturation. Above this, there is no more effect. You can inject more botulinum toxin, but the effect is, so to speak, finalized. There is a plateau. That means that is a waste of units when you inject more. You should just inject the right dose to get inhibition, but more makes no sense. So this was a huge retrospective study with more than 1,200 subjects. And in this case, the patients were treated with different products in different visits. So the treatment was divided in 2 intervals, 3 visits, and there were different groups. One group treated with incobotulinum toxin A, the pure neurotoxin, could be treated in the next interval with incobotulinum toxin A, that means with Bocotur, or with Botox, or Vistavex, or botulinum toxin A, or with Azapur. And the same procedure or the same regimen was carried out with patients who were treated in the first interval with onobotulinum toxin A. They could also switch over to the other products and so on. And so it was now analyzed whether there was a different interval. The length of the different interval was different when you compare the patients treated with incobotulinum toxin A in the first interval and the next interval with onobotulinum toxin A. Would there be a difference? And so this was the result with Inco and Ona. So altogether, no difference when you compare interval 1 to 2. Same duration of effect. No significant difference. And this is just to show you the patient's and the physician's assessment of the effect. Also, no difference when you compare Inco with Ona. The same was true when the dosage was analyzed for visit 1, 2, or 3. The dosage was around 20 units in all the cases. No difference between Inco and Ona. This was without product change. So the first interval was product Inco, and in the next the product Inco again, and or Ona, and this was with the product change. So in this case, the patients were first treated with one product and changed to the other product. In all the different studies, there was no difference in the effect. So that means the patients, the physicians can switch, take this product or that product, because there's a 1-to-1 ratio in the dosage, and there's a 1-to-1 ratio concerning duration of the effect. So that's the consequence of this study. This is just to show you, we mustn't go into detail, that the requirements in the US are different, because the FDA requires that there is a 2-point improvement in the outcome, and so the response rates are different when you compare the US trials with the European trials, because in Europe there's only a 1-point reduction necessary for response. Okay, we must not go into details. That was a study that was used for the license in the US. As you know that Zim is licensed in the US for the lines as well. Okay, so this is a large consensus paper, 2 parts by Carruthers and other co-workers. And well, they just claim that there is a 1-to-1 relationship between Bocotur and Botox or Vistadel, because there were some doubts. Elegant was claiming, well, the potency of Bocotur is not the same as the potency as Botox or Vistadel, but that was in the beginning, and now this has changed. I think that's really true. There is a 1-to-1 dose-response relationship. This is a study by Prager from last year. Only a small study. He showed that after 3 days he has more than 95% response rate, so very fast response rate. And interestingly, after 5 months, he still found 76% response. So long duration of activity in this, I must confess, very small study of 23 patients. So this is, I think, the just recently published study in August this year with the comparison of the different products, 180 subjects, 3 groups, every group 60 patients. 21 units inco, owner, and 1:3 ratio to eubotulin toxin, 5 injection points, you know that, for the treatment of glabellar lime. And this was a result concerning duration. And there is a difference in the duration of a male when you compare it with female patients. But altogether, when you compare the different products, there is no difference in the duration of effect in this study, which I think has significant results. So conclusion again, not only potency is 1:1, but the duration of effect is also 1:1. So that were some clinical studies I wanted to present to you. Maybe you have questions concerning this topic.
This was the same number of units, I suppose.
Yeah.
In your experience, what is the conversion ratio of abobotulinum toxin and incobotulinum toxin?
There are not really
there's a lot of debate in the literature of abo versus ona.
Yeah.
I have never seen anything, but I may miss that.
No, there are not. But there is one small study, and I think it was 1 to 2.5.
Yes. So
and
the same.
Yeah.
The same.
Yeah.
As ona.
Yeah, exactly. Must be the same, because when there is 1:1 inco to ona, then there must be the same ratio inco to abo, and there is ona to abo 1.5. So that's a good conclusion. But there is not a big study, but a huge study which is analyzing the ratio between inco and abo so far.
It's really interesting that we today we learned that with the Bokutur it works after 12 months also in the Sahara deserts. Is it?
Yeah.
Yeah. it's really interesting.
Okay, my last point or last issue is the LD50 assay. You know that it's an animal assay, and well, I'm not familiar with the situation in Italy here, but in Germany and other northern European countries, there is a lot of debate and a lot of attacks by animal protection groups. And they are fighting this assay because it consumes a lot of mice, I can tell you, more than 100 mice, and so there's really a fighting against this assay. So there's also pressure from the government to replace this animal assay by other non-animal assays. And there are some different approaches. One approach is, of course, to use this hemi-diaphragm assay. And Merz has invested a lot of work in this assay, but it's still dependent on animals. Just know you need a mouse to prepare the hemi-diaphragm from the mouse. And so the more elegant approach is to use cells to develop a cellular system. And you can use different cell systems, as I have listed here. You can use primary neurons, but then you have also to kill mice in that case. You can use neural cell lines. You can use neurons from embryonic stem cells, not so easy to prepare. Or the other thing is you can use neurons which are induced or prepared from induced pluripotent stem cells. What does that mean? Well, at first, there have been a lot of different cellular systems in the literature, but all these systems were not sensitive enough. When you want to replace the LD50 assay, you have to show that your system can really detect one LD50 unit. That's the requirement. That was a hurdle, a barrier. But now there are methods to spring over, to jump over this barrier, and that are induced pluripotent stem cells. And there was the Nobel Prize last year to this Japanese scientist Yamanaka, who developed this induced pluripotent stem cells. And what is carried out in this case, you take stem cells that are fibroblasts, and then you transduce a couple of growth factors into, or transcription factors into these cells, which is called reprogramming. And then you have induced pluripotent stem cells, which can develop into any other cells. And when you incubate these cells with retinoic acid and growth factors, you can differentiate these cells into neurons, which really look like motor neurons. You see these dendrites. These cells really behave like motor neurons. And these cells can be used to analyze the activity of the neurotoxin. And here is shown how it is carried out. You have these differentiated human-induced pluripotent stem cells. You seed them in microtiter plates, and then they are incubated with botulinum neurotoxin. You lyse the cells, and then you analyze the concentration of the cleaved SNAP25. And this gives you a measurement for the activity of the neurotoxin. And then you have a standard curve, and with the standard curve, you can really analyze the concentration of a batch. And that's what is this assay for. Every batch which you use in the practice must be released from the company. So every batch must analyze, has it 100 LD50 units. And, until now it's carried out with this LD50 assay, but in the future an assay like this will be used to really analyze the activity of a batch. But that is not so easy, because it must be carried out under GMP conditions. It must be a validated assay, and for quite some time you have to have this LD50 assay and the stem cell assay or cellular assay in parallel to demonstrate that both assays give you the same result. So that takes quite some time to carry out this development, but in the end, Merz will have such a cellular assay, and you will get rid of all the animal assays. Okay, that's it, what I have in my presentation. Unfortunately, I don't have all my presentations on my stick, so...
Okay, just a few spot questions. Future applications. Clinical.
Future applications in aesthetic field, we already talked about that. Crow feeds, of course. Forehead, you mentioned that, maybe problematic. We are also thinking about, for example, acne or rosacea. That could be also in the dermatological field an interesting application, but we must really analyze that. I'm not so convinced of it. My other colleagues are more convinced that it could happen, because the sebum cell is not activated by acetylcholine. And only if acetylcholine is involved in the secretion process, botulinum toxin can act. So we will see, maybe I'm wrong, maybe, and maybe botulinum toxin can also work in these, with these cells. And there is also the idea to use botulinum toxin in wound repair, that some factors are affected by botulinum toxin. That has to be shown. There are some developments, not by my company, but in other companies. I'm a little bit hesitating, because I'm a strict scientist, so to speak, and I must be really convinced that botulinum toxin has an effect on this or that cells. It must bind to the cell, it must be taken up, there must be SNAP25 in the cell, otherwise botulinum toxin cannot work. And unless I'm not really convinced by scientific data that all these components are present, I'm a little bit skeptical about that. So that is this field and maybe future fields in which I'm a little bit related, but that are confident issues, so I cannot go into detail. You know that botulinum toxin is very specific to the motor neuron and to special gland cells, which are activated by cholinergic cells. And the reason for that is that the binding domain is very specific to this SV2. So the next step, or another step for future development, and we will go into this field, is that we exchange the binding domain with other binding domains for other cells, so that we can inhibit pathologic secretory processes in other cells. For example, pain, for example, well, there are a couple of other targets which can be directed. So that's, from a scientific point of view, I'm very enthusiastic about this concept. We use a molecule which is produced by nature and change it a little bit to get a new specificity. And you can even go further. There are secondary processes which are not governed, which are not determined by SNAP25, but by other molecules. So you can also change the protease activity of the light chain and so target other secretory processes. So from that standpoint, it's a very, for me, fascinating molecule. Invented by nature, and I think this was a very smart molecule to have such a range of different activities. This binding domain, this binding process, very specific. You have this translocation, also an interesting activity, because usually proteins have no mechanism to penetrate through membranes. But these Clostridia have invented this translocation domain to transport proteins through the membrane into another compartment. That's also an interesting activity which can be used for pharmacological purposes. And well, we will do that, of course, but I cannot go into detail, of course.
I read some publication about the power of the botulinum toxin effect on some chronic inflammatory disease like arthritis. Do you know something about it? The anti-inflammatory power of the botulinum toxin?
If there is one.
For that, you need the concept which I just have explained. You need a new targeting, because the binding domain is specific to the motor end plate of motoric cells. And so you have to change that. And then you can attack arthritis, for example. Interesting project, by the way. In any case. So.
Okay. Well, this was extremely interesting. Thank you so much.
My pleasure.